Welcome to our IRA seminar. It's my great pleasure to introduce our esteemed seminar speaker today, Dr. Andrew Senior. Andrew is currently a research scientist in the science team at DeepMind in London. He famously led the AlphaFold team for the CASP 13 protein structure assessment, which brought his work to worldwide attention. Again, I should say, for Andrew has been part of some of my personal favorite papers, WaveNet and the use of LSTMs for Urdu acoustic modeling included. Andrew received a PhD from Cambridge University for his thesis on recurrent neural networks. He taught at Columbia University and before joining DeepMind, held research positions at Google and IBM. Today, Andrew will kindly talk to us about protein structure prediction with AlphaFold. I, for one, cannot wait. So Andrew, it's a great honor for me to yield the floor to you.
Thank you very much for the very kind introduction and for the invitation to speak to you today. So I'm going to talk about the work that we did at DeepMind about protein structure prediction, which has been work that we've done over a number of years and sort of led to a number of papers recently, in particular the AlphaFold 2 paper, the highly accurate protein structure prediction with AlphaFold. So here you can see the names of the collaborators on that paper, but it's a much larger team that's supported our work both within DeepMind and beyond. I'm going to talk about the protein folding problem, why we chose that, and then talk a bit about the machine learning of how AlphaFold works, and talk about some of the ways that we've made it accessible to the outside community, and then some of the impact that it's beginning to have in the biology community. And then I'll close with some sort of future work and talk about DeepMind's problems that DeepMind is tackling in science and the future of ML for science. So DeepMind, we started out with a mission to solve artificial general intelligence, but we've actually sort of reworded our mission statement a little bit to recognize that one of the areas where we think artificial general intelligence is going to have a big impact is in solving fundamental scientific problems. So that's part of our mission statement, is solving scientific problems with AI and the techniques that we develop on the road to developing artificial general intelligence. And solving the three-dimensional structure of a protein is a fundamental scientific challenge. And we're very keen on looking at these fundamental problems, not just tackling problems where ML can help with science, but things where we're solving, as we call them, root nodes, which will have many repercussions and sort of lead to a long life for the solutions that we develop, not just solving a single sort of point problem that doesn't have sort of longer repercussions. And so we picked out this particular problem as a sort of grand challenge in science that was amenable to machine learning. And then AlphaFold is the name that we've given to the system that tackles this problem. And we believe comes very close to a solution, actually solving the problem. So proteins, we all talk about protein, but mostly in the context of diet, but they're actually really molecular machines. They're these molecules. They're essential to just about all of life's functions by the actions that they cause. So they can have structural function. So hair involves protein. So it's just the sort of physical structure there that's important. But they also act as enzymes. Enzymes are catalysts in the form of proteins. So they enable a lot of the chemical reactions in cells. Antibodies, we've heard about antibodies a lot in the last couple of years. Those are a kind of protein that the body builds as an immune defense system. But proteins are, you can think of them as linear molecules. They've got this repeating structure of the amino acid backbone, and they're just concatenated into a long chain. But there are 20 different amino acids that are commonly used in the body, and the combination, as you string them together, leads to a very large space of possible molecules. So we typically work with proteins of lengths 1 to 300. They can be very much larger, and they can be very short. But that gives you a space of 20 to the 300 possible sequences that you might have. And what is known about the proteins is, to a large extent, the three-dimensional structure that a protein folds up to it determines its function to a great extent. So on the right, we can see some proteins where we've just traced out this backbone, the linear chain that repeats. It's basically a nitrogen-carbon-carbon repeat pattern that traces out this arc through space with the sort of chemical function in a side chain that comes off that main backbone that actually determines the protein sequence and the chemistry of each of these pieces. And it also determines, to a large extent, how it folds up into this three-dimensional shape. But we can roughly represent the structure by tracing out the path of the backbone in three-dimensional space. And on the right, we have these cartoon representations where we've highlighted what are known as secondary structure motifs, which are these repeating motifs. So a helix is a very stable construct in proteins where you have roughly 3 amino acids repeating in a helical structure. And then you also get these strands that form into parallel shapes, which we call sheets. And those are highlighted in this cartoon representation with the arrows. But essentially, these are just paths through three-dimensional space. But the chemistry is determined by the side chains, which are determined by this particular amino acid. So understanding the shape largely determines the function, whether a protein is going to act as an enzyme, act as a signaling molecule, act as a structural molecule. And so we're very interested in understanding what the three-dimensional shape of proteins is. So it has repercussions in all areas of biology, really. So why do we want to do this as a machine learning problem? There's a large database of protein structures that have been solved experimentally. There are techniques such as X-ray crystallography and more recently cryo electron microscopy that are used to solve the three-dimensional structures experimentally. But the trouble is that can take years to solve proteins. And there are lots of structures that people have tried to solve and they haven't managed to actually solve the structure experimentally. And you can see here some of the steps that are typically involved if you're doing, say, X-ray crystallography. And typically people say it can be a PhD just to solve the structure of one protein. So if we could do it in a computer, then potentially we could do it much faster. And we'd have a different error characteristics as well. There are errors involved in these experimental structures. Possibly with computers, we can actually avoid some of those errors and extend it to other problems that are not solvable yet by the experimental techniques. So here's another example where here I'm not just showing the backbone, which is this three-dimensional curve that goes throughout the shape, but also I'm showing the side chains. So these are short chains of a few carbon atoms. There are a few other elements that are also involved, like nitrogen, oxygen, of course, hydrogen, that determine the chemistry of these individual side chains. But largely the structure is determined by the backbone shape, and that's what we focused on predicting. And when you get the backbone, then it's easier to fill in the side chains. And to some extent, those are quite variable as well, that they will move around because they're in solution. And this is an example from a test set that we have of a protein where the structure was experimentally determined. That's the green ground truth path, and the blue is our prediction of our system. So we are predicting not just the backbone, but also the side chains. And you can see there are places where our structure does deviate from the truth. So on the right, you can see a blue loop that's just disappearing now. But to a large extent, the backbone does trace out the same path in our prediction and the ground truth. So one of the reasons why we tackle this problem, not only is it a very important fundamental problem in biology, but it's one for which there's a very clearly defined success metric. And there is a large amount of data on which to train. And there's a community that has evolved around the practice of predicting structures. So people have been trying to solve this problem for 30, 40 years using computational techniques. And they've built a community around this critical assessment of protein structure prediction, where every 2 years the community comes together and does a blind assessment of the current software techniques for predicting structure. And they do break the problem down into sort of various sub-problems, but the main problem is given a single molecule, protein molecule, where you're given the protein sequence, basically the character sequence of the amino acids that form that protein, what would its three-dimensional structure be? And just looking at the background, the backbone structure as a way of having a simple metric. And they developed this metric called GDT, Global Distance Test, where 100% would be exactly matching the experimental structure. And a background of sort of a random guess of a sort of connected chain might score sort of 20 or below. It depends very much on the size of the protein that you're predicting. So this community has organized this blind test where they take structures that are being experimentally solved, they sequester them, release the sequences to the community, ask for predictions. People make blind predictions. They just give them the sequence. They upload their 3D coordinates to the assessment, and then the assessors compare those 3D structures to the ground truth that has been experimentally determined, but is still kept secret. They collaborate with the experimentalists to keep these structures secret before they're published in this protein database. And the protein database is this very valuable resource that's been collected by the community, I think, since 1972. Anybody wishing to publish a paper about a protein structure, they're obliged to publish their structure with open access in the protein databank. So we have hundreds of thousands of structures now in this data set, which makes it very valuable for machine learning. In the bottom right, so the top left, you can see a couple more of the structures that we've predicted. One much more complex one with a lot of helices, but again, we get very good global alignment, and in a lot of places, very good local alignment between our prediction in blue and the ground truth in green. And on the bottom right, you can see the performance in gray of the people's systems in the historical performance, where over every 2 years from 2006, that was CASP 7, to 2016, the performance has been varying, sort of 30 to 40 GDT. And then we entered the competition for the first time in 2018, CASP 13, with our AlphaFold 1 system, and made a step change compared to the previous solutions. And then we went away for another 2 years, worked on the problem some more, and entered in 2020 with AlphaFold 2 with another step change that makes a big advance over the state of the art of previously and all other competitors who were entering that assessment in 2020. And here we're getting, I think this is the median, this is the mean GDT across the proteins that were assessed. I think it's about a set of about 40 proteins. And in conversation with the assessors, they basically said that a GDT of about 90 is solving the problem. So our median GDT is above 90 GDT. We're very often getting numbers above 95. So the assessors basically categorize this as solving the problem. We're never going to get perfect agreement with the experimental technique, partly because the experimental techniques themselves have their own noise characteristics. They're limited in resolution. And like crystallography, there are packing artifacts from building up a crystal of multiple proteins that sort of the edges interact in a way that wouldn't be natural in solution. And proteins are inherently flexible structures, some more so than others. So there is some inherent variability in the structure. There's no single set of coordinates that is exact truth that we can achieve. In some cases, there may be multiple structures that are achieved, that are embodied by proteins. So things like pores, they may open and close in cell membranes. So they have two structures, say. And the experimental data may only contain one of those structures. And sometimes experimentalists find a way to force the other structure, and they can measure two different structures. And our system, we're treating it very much as a supervised learning problem, where there is a single true structure we assume for every sequence. And so we come out with a single set of coordinates. And in practice, that's one of the sort of frontiers, is we would actually like to understand the distribution of structures. And that's one of the sort of grand challenges that's still open, where we make a single point prediction, but we would like to really understand the flexibility and the full distribution of the landscape of proteins. So now I'm going to go on to talk about how AlphaFold works. We published our method. We disclosed our method at the time of the competition. There was a conference where we gave a talk where we described our method in a talk. And then we published this paper in Nature in 2021 with very detailed description, the pseudocode for all the operations, with all the sub-algorithms that fit together to make AlphaFold. But we've also released the code. So for AlphaFold 1, we released the inference code that was just enough to replicate our results, to sort of know that we had done how we had generated the structures, but we didn't release the actual featureization of the proteins that was essential if anybody wanted to put their own protein into our system. But that's what we've done with AlphaFold 2. We've released the code, and we've released it in a collab, which is a very user-friendly way of accessing the code, so anybody can run AlphaFold on their own proteins. And we've released the full weights of the neural network, as well as the code for inference. So before I go into talking about the details of how it works, then just a summary, which I think you'll all be familiar with, about the idea of inductive bias for machine learning models. So really, a lot of the process of building AlphaFold has been trying to understand what the structure of the problem is, and then trying to embody that in the neural network. So the sort of standard zoo of machine learning algorithms, there are these things like convolutional networks, where you repeat the same operation at many locations, say across an image, where you want to be able to recognize, is there a dog in this image, regardless of whether the dog is in the top left-hand corner or the bottom right-hand corner. So convolution provides a very good model for thinking about that position invariant prediction problem. Recurrent networks are very good at processing sequences. Say in speech, we want to do speech recognition. We want to recognize a word regardless of when it is said. So we want to basically apply the same operation at every point in time. So convolutional network across time, or a recurrent network across speech or across language is a very good model. Those are good for one-dimensional signals in particular. Graph networks is another paradigm for data which has a graph structure, particularly molecules. And attention is the sort of the very popular paradigm now in transformers, where you can process an unordered set, but the network is able to dynamically focus its attention on certain features, certain areas of the input, and build up a representation in a more fluid manner. So we've combined elements of these systems, but particularly attention, which with transformers has sort of transformed nearly every field of machine learning in recent years. But we particularly want to take insights from the physical, and in this case, geometrical problem of protein structure, so that the network reasons in an economical way about the data that we give it. If we had unlimited data, we could construct a model that might learn very simply, like a very simple multi-layer perceptron might learn with infinite data. But we have this limited resource of these few 100,000 structures in the protein data bank from which to train. And throughout this process, we've questioned whether that would be enough data to train the model. And we even at the beginning considered, what would it take to just buy more data? Can we just, buy an X-ray system and start collecting data. And that we very soon realized that it's a very difficult process. It's this very time-consuming and expensive just to get one protein, especially if you want something that's different from the ones that are already in the protein data bank. The protein data bank, PDB, is very redundant. It has hundreds of thousands of structures, but when you take out all the copies of the same protein with different small molecules added in or crystallized under very slightly different conditions, then there's actually very much less diversity than you would expect. It's more like maybe 100,000 distinct things when you cluster them with to reduce the duplication. So we want to build these geometric insights into the model, and we want to get an end-to-end system that directly produces a structure. So AlphaFold 1 processed proteins and predicted attributes of the structure, and then used a gradient descent technique to actually fit those attributes, fit a structure to those attributes, and resulted in good predictions, but it didn't do it end-to-end with a neural network. So we had this disjoint system that it was hard to optimize the whole thing. But with proteins, we have these physical constraints. So one, we know that the protein is a chain. So in fact, you know the distance between the atoms on the backbone. Those are covalent bonds. You know the distances of them. You know something about the geometry of them. But there are a lot of free parameters, and we want to sort of understand how we can encode other constraints about the model. And one of the things that had already been appreciated before is that when the protein sort of folds back on itself, certain residues, it's another word that we use for amino acids, so we have a sequence of residues, certain of these residues come into contact with each other. And there are certain things that are favorable or unfavorable, like having a positive charge, it wants to be next to a negative charge. You can't have two positives close together in a protein. So there are constraints about which residues are going to be in contact with each other. And in particular, the proteins typically fold up, they're in water, but they want to fold up to hide a hydrophobic core. So there are these residues that don't want to be next to water. They try to fold up in the center of the protein where the water is excluded. And then the residues that are left on the outside are the hydrophilic ones that like to be in a watery environment. So you can start to build together some of these constraints, and people had already worked out some of these to understand what structures are likely. But still, the space of protein structures is vast. So it's very hard to put together enough constraints to learn about the structure of a protein. But we talked earlier about graph neural networks. The structure of a protein can be seen as a graph. The trouble is that that's the output of our network. So we can't really use graph processing as such because we don't know what the graph is. But we can see this problem as a graph inference process, where we want to work out which residues are going to be close to which other ones. And we can say from the beginning that, say, two positive residues are unlikely to be close to each other. But a positive and negative residue are more likely to be close to each other. So we can, the AlphaFold 1, we built up this idea of a probability distribution of the distance between two residues, extending previous work where people had looked at the probability that two residues would be in contact. We actually started to predict these distances. And that's a side effect of the current model. But we also then use have extra layers of the model that use these constraints that we're building up about sort of, it's a probabilistic graph. Are these things likely to be in contact? How close are they likely to be to each other? And it reasons about that graph and then builds up a structure that satisfies that graph and the other properties of proteins, like having the hydrophobics on the inside, the hydrophilics on the outside, and fitting this space-filling curve. One of the fundamental features that we use for making this prediction is one that had been established by the community over the last 20-25 years. People hypothesized the idea that because proteins are descended are encoded by genetic sequences, And if we look at the tree of life, we have common ancestors with other species. In general, we will have similar proteins to that ancestor species. And other species that are descended from that common ancestor will have similar proteins to us. And by the process of mutation, then those protein sequences will change over time. And only mutations that preserve fitness or improve fitness will be preserved. Mutations that destroy the function of a protein will cause an organism to die out, quite likely. So we can use the evolutionary history to make inferences about the proteins. In particular, what we do is we look at, if we're given a protein sequence, then we can look across all the other protein sequences that we know of from other organisms and look for related protein sequences. And we have very large databases of protein sequences because of genetic sequencing. Especially over the last few years, the number of organisms whose entire genomes have been sequenced has grown very, very rapidly. And from just the genetic sequence, you can infer where the open reading frames are, the pieces that are going to code for actual proteins. You can actually infer where those are, and you can know what the protein sequence of an organism is without ever having actually observed that protein. And in fact, you can do this from environmental DNA. You dip a bucket into the sea, sequence all the DNA you can find without even finding the organisms necessarily. And you can just infer where the proteins are in that sample. So we have actually billions of known protein sequences, often for organisms we don't even know, for proteins that we've never actually observed, but we believe they do code for proteins somewhere in the wild. And for those, we don't have any idea about the structure. The structure data that we have is limited to this PDB data set, which has been experimentally determined. But if I'm given a new protein sequence, I can then look in these very large databases of sequences and find other protein sequences that are similar. And typically people talk about sort of 30% similarity means that a protein sequence is going to be related, because the protein space is so large, the chance of having 30% similarity in a sequence of 200, 300 residues, that's not going to arise by chance. That means that these two sequences have come from some common ancestor. And then we can look at the similarities and say, the mutations are random, but things that are conserved in those sequences are going to be things that are critical for function and structure, ultimately from structure. And we can also look at what's called coevolution, where things that vary together are likely to be in contact. So if we look, this is a sort of diagram showing the sequence going left to right, and then a set of species going top to bottom. So we've aligned these sequences using a sort of edit distance alignment. And we say, if we look at particular locations, we've got our human protein at the top in this case, where that's our query protein. We look at all the other similar proteins and we look at what happens. So in this shark DNA here, we had the same green amino acid and the same orange one and the same red one here, but the blue one turned into a red one in this particular location. But across all these sequences, we had this orange Y as we've represented it. So that's been preserved. It's likely to be very important for the structure or function of that protein. And the others to a lesser extent. And we can also look at when the protein folds in on itself, pairs of residues that are in contact, they have to be consistent. So in this case, we've got these sort of cartoon shapes that fit together. That could be charge matching or both being hydrophobic matching. So we need consistency. If the red Y mutated randomly and turned into another green blob, then in the left-hand structure, that might make the protein unstable and unable to function. So that creature is only going to be able to survive, or that lineage is only going to survive for a long time, if a corresponding mutation on the other side, in that case turning from a green blob into a red Y, counteracts that imbalance. So you get these pairs of residues that co-evolve, that if you make a change in one, you're likely to have an opposite change in the other one, or some kind of balancing change. And over large sets of sequences, then we can find these correlations and use those to infer contact. And as we showed in AlphaFold 1, you can actually start to infer the distance based on the statistics of these pairs. So we built on prior work where people had originally hypothesized this, then they'd done the maths to show that you could compute statistics that would indicate where there was likely to be a contact. We started to predict distances, and then we fed that data into our AlphaFold 2 network, where we just give it the raw sequences. We give it these sequences of which amino acids line up across a set of species, and we let the network compute the statistics, rather than relying on classical statistics to say, is there a correlation here or not? What's the probability of a contact? We just give it as raw data for the network for it to infer what it can from this data. So going to our network, we're given an input sequence. We go and search in a genetic database and find the multiple sequence alignment of all the similar sequences that we can find that are similar to this sequence, and we line them up so that the corresponding amino acids are in columns, possibly with insertions and deletions and substitutions. And then that's part of the data structure that we present to the network. And we know that multiple sequence alignment is very useful for inferring structure from our earlier work. But we know that a pairwise representation is important, that knowing the distance, that whether these residues are in contact or the distance between them is going to be important. So we use the inductive bias of constructing a two-dimensional representation. So for every pair of residues, we're going to have a piece of the neural network that represents what we know about that pair, so that we can make a prediction about are these in contact. And basically, in the graph inference version of this problem, are these a link in the graph, or are they disconnected? Another standard technique that's been developed over the years is to know that because these proteins are very often related to one another, if anybody has solved a structure of a sequence that's similar to your sequence, then chances are that the structure is going to be very similar. So it's been really almost a separate technique of looking for a template structure, one whose structure has already been solved, and then adapting that structure to match your sequence, to say, oh, there's an insertion here. How do we fit that insertion into that template structure? Or this residue changed, how would that change the structure relative to the template? In our first system, we didn't even try to solve this problem. We thought that's already quite a good, that people already have quite good systems, and they produce usable results. So we were tackling the hard problem where there isn't a template. But for the AlphaFold 2 system, then we did add that information in. And if there is a template, we provide it. And because it's a machine learning system, and we don't have to actually have a sort of physical model that we're adapting, we can actually provide remote templates where we think there's a similarity, but we're not sure. And we feed it into the network. And over training, we learn how much of that information to use and how to adapt that template for the final structure. But when available, we do provide that structure. And if it's available, then it's likely to provide a boost to our performance compared to if we didn't have that. So going forward, we have these inputs, and basically we have a representation that's based on the multiple sequence alignment. So this is a two-dimensional array with residue index or position in the sequence horizontally and species vertically. Then we also have this pair representation, which is just residue index in both X and Y, and a given cell represents the correlation between those two residues. And then we have a network that processes this information. We call that network EvoFormer because it uses the evolutionary information in the multiple sequence alignment. And we update these representations, put them out at the end, And then we have a structure module that takes that information and creates a set of coordinates. And as part of this, we built an attention mechanism that works in 3D space. It's rotation invariant and allows us to produce molecules without having to know their orientation. And that was an important part of getting to this process. But in fact, it's not an essential part. Having built the rest of the structure, then we find that we can use much simpler techniques to generate those coordinates at the end. But having this three-dimensional reasoning between residues was very important in the first case. Another important factor was looking at the losses that we're going to use to train our neural network. So we're going to do gradient descent to optimize an objective, which is minimizing the loss of how mismatched our predicted structure is to the ground truth. And we designed this, what we've termed the frame-aligned point error, as a rotationally invariant loss metric. And in particular, one thing that we realized for proteins is they've got this backbone structure with the nitrogen-carbon-carbon sequence, where those three atoms form a fixed angle. So you've got they define a two-dimensional plane, or as we think of it, they define a rigid frame in three-dimensional space. And those are all going to be similar. And in particular, we can use those to align the predicted frame of one residue with the ground truth frame that corresponds for the same amino acid in the ground truth. So we do that one residue at a time. So we pick the nth residue, and we have it in our predicted structure. We have that same nth residue in the ground truth, and we align the molecules only using those three atoms. And we can do that pretty much perfectly because the geometry is fixed. And then we can look at the errors in all the other residues. So here on the right, we've taken the true frame in green, we've aligned it with one of those predicted frames. So there's a perfect alignment in the middle. And then we can look at the errors in the frames, in particular, in this case, the ones that are nearby. We can say, oh, this one needs to be twisted a little bit and slid over. So we've got a rotation and translation component to the error. And we can also frame that as a distance error in each atom. So that's basically how we score the predicted structure at the end. And that was an innovation of how to construct a loss that is rotation invariant and is dependent only on the alignment of an individual frame and can work even from the very beginning where we have complete spaghetti structures being generated. And also another innovation was the idea of predicting these frames completely independently. Although we know a protein has these frames lined up next to each other at a fixed distance from one to another, we let the network predict them anywhere it wanted in 3D space, rather than forcing it to predict a chain. We know that the protein chain is this huge constraint that we do ultimately have to satisfy. But we allowed the model to predict those frames anywhere in space and just enforce the constraint that they will need to be close to one another with a component of the loss. Rather than, like in our first model, we had a model that was forced to produce a chain, and then it just had to adjust the angles within that chain. And that's very difficult. It's a very ill-conditioned problem because you've got this chain of rigid links with certain rotations. It makes it very difficult to adjust that to match your final structure. But this idea of predicting frames in three-dimensional space and just letting the model slowly move them into the right location because that doing so will satisfy the loss proved to be a very useful way of generating proteins. It was different from our original thing. So if we look at our network, we've got the inputs on the left. We turn them into these multiple sequence alignment and residue pair representations. We feed it through this Evo former, produce a structure, and then we have this concept of recycling, where we feed the same structure back in at the beginning of the network, and then let the network refine it, basically, make an improvement. And we do that a few times, and we vary that number of times in training, and we find that this improves performance, that we've trained the network not only to predict a structure from scratch, but also to improve a structure if it's nearly right. It gives it more chances to improve it. Another aspect that we found was important, that was an innovation from our original model, was the idea of predicting confidence. So we've basically regressed accuracy measures that we see from our prediction. So we make a prediction, we measure how far it is from the ground truth, and then we train an extra output of the neural network to regress that accuracy measure. So we have this per residue confidence measure called LDDT. LDDT is an error measure that was already established in the community. And it basically says, how accurate is this one residue relative to the rest of the structure? And here we've got a color-coded graph. Very high is blue, very poor is red. And we can make it, we find that these are actually very well calibrated. So when we make a prediction, we can actually know which bits we're confident in and which bits are not, and possibly an entire structure we may not be very confident. And then we can make a pairwise version of this, a predicted aligned error, where we predict whether we have the relationship between two residues correct. And from this, it's a two-dimensional version. We can look at blocks on the diagonal. Those are pieces where we think we have that structure consistent. We've got all the distances between those pairs of residues in a block consistent, in this case in dark green. We think we've got that block correct. But areas where it's pale, then we haven't aligned those two, the corresponding two blocks. So we'll come to that a bit more in detail later on. So for the predicted LDDT, it's per residue, so we can color the molecule. We've got these blue residues, which we have high confidence, we predict that they are highly accurate, and basically that's a confidence measure of this piece of the structure we think is good. And then we have these other pieces in red and orange where we're not confident. We think, you know, we've predicted an actual coordinate, but in practice, it may be something else. And in fact, we get these stringy versions of proteins where they're big pieces that are not accurately predicted. And people have studied these predictions and have shown that they are actually a very good predictor of whether a protein is disordered. And we find that in the body, a lot of proteins don't actually adopt their three-dimensional structure. There are proteins that are just sort of strings of spaghetti that float around, and in particular ones that only form a structure when they're in contact with a partner protein that forces a particular structure. And otherwise, they're intrinsically disordered, we say. And the people who work on disorder prediction have taken AlphaFold and looked at our PLDDT metric and shown that it's better than the previous predictions of whether a protein is disordered and which pieces of a protein is disordered. So that was a sort of an unexpected side effect. You know, we could see that this probably correlated with disorder, but it's actually been shown to be a good predictor of that. And this allows these our predicted structures to be much more interpretable, that people can trust the blue pieces, they can know that that's very likely to be true. And then the red and orange pieces, either there is no real structure or it's likely not to be true and shouldn't be trusted. You know, you need to make an experiment to verify this piece. And you know, just by looking at it, you can see that this isn't going to be a real protein structure. So I'll go back to the Evo format, talking about a little bit about how that works. And basically, it's based on attention, based on the transformer papers. We use attention to update all these elements in the representations. But we've also got a feedback across attention between these two representations. So we can have attention to update from the two-dimensional representation. We can update because it corresponds to individual residues, which are also represented in the MSA structure. And then we can form an outer product of this sequence by species representation. And then we can average over the species dimension, and then we can get a sequence by sequence representation, which is what we have in blue. So we can feed back from the crossover between these two different representations to let one update the other. Then we also use what we call triangular attention to update that two-dimensional representation, which is a process to do this graph inference, where we know that we have these pairs of residues, and because they're in three-dimensional space, they're going to satisfy this triangle constraint, that, like the triangle inequality, and in particular, these things are going to form a triangle. So if we can update these cycles to be consistent. So we build up a mechanism whereby we combine the representations of the red, green, and yellow, which each correspond to one square in this representation, so that the network can make them consistent and reason about the structure of this graph. Another thing that we did was what has been termed noisy student distillation, or it's basically self-training, where, because we know there are billions of proteins whose structure we don't know, but we do know the sequences. But we do know that they should fold, most of them are going to fold up to real proteins. They're not just random sequences that are meaningless, that we could just generate with a random number generator. Those are things that have been preserved by evolution in real organisms. Some of them may be disordered, but we expect most of them to fold up to structures. So having trained a reasonably good model at the beginning on the known structures, we then did inference on this large data set of unknown sequences of unknown structure. And we used our confidence measures to filter out the ones that we think, oh, this is disordered, it's not a real structure. But we kept the ones that we were confident in and then added those to our training set. And this, it could be an unstable process, but if your original model is good enough and your confidence measure is good enough, then you get a sort of a virtuous circle, and you're adding more structures where the sort of combination of constraints has actually led you to produce a structure that you can trust, that is useful for training. And we did that, and this increased the size of our data set many fold, and allowed us to train on more structures. So I'll quickly go through these error measures. I've talked a little bit about the predicted LDDT, which is the per residue coloring. But we also have this aligned error, which shows which pieces of a protein are consistent. And you get these domain structures, like this protein in gray, where there are two close to rigid pieces, but they're flexibly linked. And we can see that from this predicted aligned error, where we get a block diagonal square, which says the first 375 residues, we're pretty confident that we know how those relate to each other. The last chunk of 320 residues, we also know how they relate to each other, but we don't really have a very good idea of how the two blocks relate to each other. And this allows us to split proteins into reliably understood chunks. And that's, again, a very useful tool for biologists to use. So that's a summary of the machine learning. We released the code. You can use it as in a Docker container. You can use it as a collab with a sort of literate programming where you get to visualize the predictions. But we've also made a very large set of predictions. So we took a set of model organisms. These are things like fruit flies, yeast, and so on that biologists study in particular, and have released predictions for all the proteins in these model organisms. We've uploaded those to this data set, data store run by EMBL, which is a big biology lab, European biology lab based in Cambridge, who already hosted a lot of protein data. They now host our predictions and have made this available for the community to use. And we're working on increasing the size of this data set. We've made, I think, already 2 or 3 releases of increasing size, and we intend to work up to this very large data set of loose, gently deduplicated protein sequences, which would have 100 million structures, which is compared to the previously available data sets of 450 highly redundant structures that were in the PDB. So this is a vast resource available for biologists, and people are already starting to use it. Not only people who care about just one particular protein, they were waiting to see what it looked like to make inferences about the structure. But it's had bigger repercussions, both. So there are also these people who've worked on X-ray crystallography, and they collect a set of data. They work through all the long-winded process of getting a crystal, and they get data, but it's in the form of a diffraction pattern, and they need to phase that diffraction pattern to actually interpret the structure. And that's not a simple operation. It's not very well understood. If you have a guess, though, it becomes easy. If you have an accurate guess, and people try that with, they try guesses of what the structure might be to see if it fits the data. But AlphaFold now gives them a really good guess. And so we found a lot of people have been able to solve old data sets that they collected because our guess is good enough to solve this phasing problem. And then it sort of collapses into a solution where, yes, clearly the data is consistent with the AlphaFold prediction, and then they can make refinements. They can make it slightly different to our prediction, but they could only do that with a good guess. We've also seen that disorder prediction is a problem that AlphaFold can help solve, and it's being incorporated into other tools where not just this EMBL data set, but people are using the AlphaFold structures. People who study particular organisms or particular diseases are making our structures available and our predictions. So I'll just finish with talking a little bit about machine learning for science, which is sort of where we have this group at DeepMind working on scientific problems. And in the last year or so, we've actually sort of a lot of this work has come to fruition that we were doing before without talking about it very much. But we now published a string of papers in a variety of areas of science, from quantum chemistry, mathematics, using maths to guide how mathematicians work and come up with hypotheses, genomics to predict gene expression. We did glassy dynamics to understand how glassy systems move. There's recent work on controlling nuclear fusion reactors and predicting weather with nowcasting. And there are lots of other areas that we're starting to be interested in, where, again, we're trying to solve these root node problems, which will have bigger repercussions. So quantum chemistry, we can use that. If we get good at that, we can use it for designing materials, for designing catalysts, and ultimately, possibly, to help us understand the biology that we're also understanding with proteins. Similarly, genomics, that affects gene expression, which is which proteins are going to be expressed in the body. So there's a lot of exciting work in the future. We're predicting sort of extending the AlphaFold to work with complexes. We can also look at other molecules that the proteins might interact with, like DNA and small molecules. Lots of people have taken our tools and are incorporating them and building them into other tools. And we see that there's great potential for AI and machine learning in science as a whole, both in what we're doing so far, where we're building these very targeted, highly optimized ML systems to solve one particular problem, but the root nodes, which will have big repercussions. So things like prediction, generalizing from an understood area to a greater set of data, and then also providing interpretability, as we did with the glassy dynamics, say. But ultimately, with more advanced AI techniques, then there are people who are starting to begin thinking about designing AGI scientists, where you make hypotheses by reading the literature and then testing those hypotheses. That's something that we haven't worked on yet, but ultimately you can see that AGI can impact science in those ways. So I'll finish just by saying thank you to you for inviting me, and to all these people who collaborated on the paper. The community is a large, particularly CASP community who constructed this benchmark and the assessment, and the experimental biology community who provided the data over many, many years that enabled this work. And then I've got a little bit of time for question. Sorry.
No, not at all, Andrew. Thank you very much indeed for an amazing talk. And there is a sort of simple measure to see how much interest you're garnering, because we have a full cache of questions. So I'll start with that. First one comes from our very own David Kreil. Very impressive. What are your worst scores, and for what classes of proteins? I think David is referring to the measure, the GDT.
GDT.
Yeah, sorry.
Yeah,
do you understand why these are harder? Sorry.
Yes, I mean, there certainly is a range of scores. You know, our median score is in the 90s, but there is a tail. In particular, we're not as good. We're very reliant on this multiple sequence alignment data. So if you have an outlier that you can't find any related protein, then it can be hard to predict that structure. And you know, yes, we could generate sort of any GDT score potentially. And you know, some of the proteins, they may have been solved in one particular conformation and we predict a different one. So there is a distribution. It's very highly distributed up towards the you know, bunched at the high end of the scale. So our median is in the 90s. But yes, we could generate poor ones. One thing that's been found as well is that for designed proteins, so these are ones that have been proteins that have been engineered, they have no relations in evolution because they've been human constructed. They actually tend to be constructed to be very stable, and therefore we actually make quite good predictions on those proteins we've found. But that may be less true as people come up with more exotic proteins. Yeah.
Very nice. Thanks very much. The next question, I believe, refers to slide 12, and I think Yusuf was asking what should be in the diagonal. I think you may have actually later explained that. But
yes. So in this is sort of an abstract representation. But if we actually look at the distances, say, then the diagonal would all be zeros, and then most things would tend to increase as you got further away, except where the protein folds back on itself. But yes, especially for a distance, then we would expect a symmetric matrix. This is just a sort of an abstract representation. And then we predict both distances on this two-dimensional space, but we can also just have this abstract representation, which is directed. So it isn't necessarily symmetric. It could be anti-symmetric in some ways.
Thank you, Andrew. Another question from David. Have you considered structural alignments of initial structural model predictions rather than AA sequence alignments instead or in addition?
Yeah, so I mean, in a sense, that's what the templates give you a structure. I mean, you know, we don't in general have a structure as input. So structure for us is the output. We do the alignment, you know, our input space is all sequence based. But if we have a structure with a template with a structure, then we can provide that, but we align it by amino acids. But when you make these predictions, you also do want to look at, you know, is there a related protein that I can tell by the structure that's adopted? And we do look at when we do this inference on very large sets, we can look for proteins by structure. So if we know, you know, hemoglobin looks like this, is there anything else that looks like hemoglobin that we can tell because it's structured like hemoglobin, rather than necessarily that its sequence looks like hemoglobin? And you can find more distantly related proteins by that structure-based search. We definitely do that, and other people in the community have done that.
Very nice. Thank you for your answer. I have a question from Yeon Kim. It pertains to slide 19, I believe. And he says, we know that multiple sequence alignment take a long time, and we were wondering how long it took for the input to process.
Right, so I don't know the details on that. I mean, it can take a long time. So it can take of the order of minutes. You know, we have put a lot of work into sort of optimizing that, both in terms of optimizing the code and parallelizing it so that we can do it quickly. But mostly we use standard tools for this multiple sequence alignment. But for our training set, we've amortized that we cache the results and we've got it stored on disk for our training set. But if we have a new protein, then we do have to compute it, and that can take, yes, minutes, which is much longer than the network, which takes of the order of a second to do inference once it's got the input data.
Yeah, very interesting. Another question from David. Do you have a feeling whether your regions with low confidence are good predictors for truly disordered regions, say, as assessed by NMR? These are functional interest in their own right, and low informational sequence complexity is not necessarily a good predictor of biological relevant disordered regions.
Right, so this isn't something that we've really done a lot of study on ourselves, but there has been the people who work on disorder prediction and curate the disorder prediction data set, which is benchmarking that. They took AlphaFold and used our PLDDT measure. And my understanding of that work is that they said it's better than any of the existing predictors. And they've incorporated it into their online tool as a disorder predictor. But I don't know the details, and particular agreement with an NMR, whereas mostly we train on crystal and cryo-EM structures rather than the NMR.
I have another question from Youssef. Do you train AlphaFold as a language model for proteins?
No, so it's not trained that way. I mean, it has things in common, but it really is a sort of feed-forward network. It uses techniques like BERT, which is used as an unsupervised training. So we mask out pieces of the multiple sequence alignment, and we have a head which predicts those. So we're definitely using techniques in common with language models, but I wouldn't call it a language model. Like, it's not autoregressive as many language models would be. So.
Very nice. A question from Alexander Grusha. Thank you for the great talk. Could you please comment more on the disorder regions? As you have mentioned, PLDDT values are usually low, those types of regions, and is this due to the fact that there are almost no such protein regions in the PDB database?
Right, so that's true that we don't have data for those regions. Like, if you had a crystal structure, it would miss out those regions. You would just have missing coordinates for that section. So we're not training on that. We did train a head that says, is this available in the structure? And we tried to predict that fact. But we're just using this PLDDT, which is regressing the actual error measure. And it turns out that it is a good disorder predictor. But that sort of only come out after the fact. We used it as our filter for whether we train on a structure, should we trust it for this self-training. But yes, I don't know really the details about disordered proteins very much.
I love how there's so many influences that this work will potentially still have in the future. Also in that direction, Alexander actually continues. Also, IDPSs sometimes do form a structure. Do you think that it will be possible in the future to correctly predict the structure of those types of regions when they do interact with other molecules?
Yes, I think ultimately, I'm quite bullish on the idea of machine learning making these predictions, in as much as there is a given ground truth. We've released a sort of sequel to AlphaFold, which we call AlphaFold Multimer, which predicts the structures where you have more than one protein chain, where they come together in a defined structure. And people have made predictions for like how peptides bind to proteins, and to some extent how these we may make this sort of spaghetti mess for this protein by itself, but with the right partner we might get the correct structure. And so some of that is definitely starting to happen, but yes, there are limits on what we can do.
Very cool. Alexander has a final question here. Could you please explain how the PLDDT metric is computed for the protein for which there's no known ground truth?
So the LDDT is defined on the ground truth, and that you do an alignment, and you look at the errors in the distance predictions. Your structure, it said the distance between these two things was this much, but in the ground truth it was this much. So you can compute a metric based on that mismatch. But PLDDT we use to regress the actual LDDTs that we measure. So we make a prediction, we have an actual structure for something where we have the ground truth, we compute the actual LDDT, and then we have a prediction head from our network that regresses that number. And we do it as a distribution quantiles on that distribution. So when we get a new structure, we can make that inference from that head and predict a PLDDT, but that is different to the LDDT, which is only defined for a ground truth structure.
Understood. Thank you for explaining that. A comment question from Pedro Haruso. Amazing work. Did you measure the boost of using the noisy student distillation?
We did. I couldn't quote you any numbers on that. I mean, there are numbers with a lot of ablations in the paper, in the supplementary materials, where we take out various pieces and show what matters. And we were kind of surprised how robust the technique was to taking out some of these components. That things that we thought were absolutely essential for this method to work early on, we could take them out at the end and do something much simpler or not have it. And the system would, it would lose 2 or 3 GDT for something. So I'm not sure. It all combines to get that top performance, but I don't think it's a huge difference. I think a system trained only on PDB performs quite close.
Understood. Yeah. Another question from Youssef. He says, what's the meaning of the coloring in the proteins? It looks continuous, but the protein is not. I'm not sure which slide Youssef might be referring to.
Yeah, so this one, it's colored by this PLDDT. So each residue is colored blue if we're confident, red if it's not. I think that's the only real coloring that we used. And the proteins, usually they are continuous. Sometimes the ground truth structure is discontinuous because the crystal didn't resolve certain residues, so they have gaps. But our structures generally are continuous, although it's not constrained to be. It's just we've encouraged our model to produce things that have this continuity. But it's really, that's kind of a function of the graphical tool, that how it renders it, whether it chooses to join things together or not in the visualization. But also the coloring elsewhere, there was blue for our prediction and green for ground truth. But, I think that's the only coloring scheme I can remember.
Nice. Another question from Alexander, which is how many sequences and to what degree do they need to be similar in the MSA for the AlphaFold model to provide accurate predictions?
Right. So that's something that we've measured, like how many sequences do you need in the multiple sequence alignment? And that's something that we were able to push down compared to AlphaFold 1, getting to AlphaFold 2, then that number has gone down to something like 5 to 10 sequences is enough to give us a good structure. Whereas before we would talk about 50, 100 sequences of similarity. But it's very much a continuum of how do you define a similarity, because you may find 50 proteins that only change by 1 amino acid, because they're very closely related organisms, or somebody did an experiment where they mutated that 1 amino acid. So diversity is very useful. We cluster these sequences, so we have a measure of how many sequences after we clustered them. You may find thousands of sequences, but most of them are slight variations on each other. That may only count as 2 sequences, say, if they're clustered together. But yeah, if we've got 5 to 10, then that's when we start really doing much better.
Understood. Alexander has another question. Could you please comment more on the prediction of protein complexes? How far is AlphaFold now from being able to predict them?
So we've released an archive paper and the corresponding code with this AlphaFold multimer. where we predict certain classes of protein complexes. I mean, there are these huge complexes in the body, like the ribosome, which is used to actually construct the proteins in the body. So it reads in the RNA and it produces proteins. That's a big complex of both multiple proteins and RNA. So, to be able to predict that, we're sort of a long way from being able to do that task, where you've got a lot of components, they're of different molecule types, and they're very flexible as well. They move around. It is a machine. But for small complexes where maybe you have just 2 or 3 protein types and they fit together in a sort of regular geometry, or you've just got 2 proteins, how they fit together, then we're starting to get pretty good results on those. But it is a more challenging problem than just what we focused on so far. And yes, there are also these viral capsids where they have hundreds of proteins that form this geometric array, where you really want to have a very specialized technique that understands the symmetries involved. You've got this very nice rotational symmetries that you can exploit, but our system had limited understanding of those big symmetries. We've focused on the small complexes so far.
Very nice. I have a question from Ben Gruten. A great presentation, thank you. Interesting to hear that AlphaFold 2 also trained on some data that it labeled itself. 2 questions that I have about this. Number 1, what proportion of the unlabeled sequences did you end up labeling with a high confidence and thus use for the training? And number 2, what confidence level did you deem high enough to add the sample to your data set?
Yeah, so I don't remember the actual numbers that we ended up using for the system that we released, but it's something, I think it's something like sort of 70 LDDT for enough residues. So it has to be pretty high confidence. And we throw out a large proportion, I think. I mean, partly because we can generate, there are billions of sequences ultimately, although when you cluster them, it comes down much smaller. So we spend inference time to make predictions, but we can afford to throw a lot away and still have a lot of proteins left over. That's much bigger than our actual experimentally determined training set. And we don't mix in too much of the predicted structures because they're less trustworthy. So we still keep quite a high proportion of the ground truth of the experimentally determined structures in the training set. Even though we have a much bigger set of predicted structures, we don't sample very often from that pool.
Very nice. Yusuf would actually like to add to that question. Can unsupervised training on unlabeled data help make the model more accurate? Thank you.
So, I mean, this self-supervised training, that definitely helps. That's what this noisy student distillation works. And to me, that's a process of, if you have enough different constraints coming together and your model is sufficiently accurate, then you can use that. We saw the same thing in speech recognition, which I worked on before, that when you cross a threshold of accuracy, then you can start doing this self-training. But I think unsupervised training, like the BERT objective, I think, again, that doesn't actually add very much to the system in the end. So the unsupervised training is not that useful. And we've really, focused on just the structure-based objective. And I think it'd be hard to get much out of the unsupervised training. Although a lot of people have done that for protein sequences. They built unsupervised models, just of language models of protein sequences, but they tend to be for other objectives than structure.
Fair enough. Thank you. A question from Madi Hasselbankori. Amazing presentation. Thank you. In case of proteins like ion channels with multiple functional states, which are open, closed, desensitized states, is there a possibility to use an experimentally solved state as constraints to prepare the model of the state with AlphaFold ourselves? To use AlphaFold instead of conventional homology modeling as the available predicted AF, so AlphaFold, I guess, structure for my target does not exactly correspond to any of the 3 known states.
Yeah, so that is a problem, and we ran into that in the competition, the CASP assessment, that there was a structure where we felt, okay, this is going to have two states. Can we trick AlphaFold into giving us two diverse answers? So we actually did. We ran multiple models. You're allowed to give multiple guesses or predictions in your entry to the assessment. So that gave us some diversity by just having different models. But we tried putting different templates in to try and bias the network one way or another. And there is, I mean, people are doing, there's a lot of AlphaFold hacking out there in the community of people taking our model and trying to trick it in different ways to produce things with desirable properties. So you can give it a template. You can hand construct a template to try and bias the predictions one way or another, possibly. And we did try a little bit of that, but really the ultimate goal we want a generative model that will give us a distribution over all the states. But that's a very challenging problem. That's going to keep us busy for many years to come.
Great. Question from Mohit Roy. Great work. I was wondering, number one, how much difficulty you had with beta sheets in AlphaFold 1 and 2 because of the implications of RNA structure prediction. And a second question he had is if you could elaborate on how you trained over the 3 cycles of residues. Thank you for your presentation again.
Right. So with beta sheets, I mean, we didn't do anything special. I think that's something that definitely there was something that improved from AlphaFold 1 to AlphaFold 2. In our very early attempts, it was very easy to produce helices. They're very natural structures that there are sort of local energy minimum that is very easy to predict for any protein. But sheets, getting those to line up, it's not something we did anything special for, but it now comes out of the overall model being good enough. And then for the 3 cycles, it's a little bit complex to explain, but if you've got a pair ij and jk, so basically those are elements in that pair representation, then you can combine those representations to produce a representation for ik. So you update ik based on the elements ij and jk combined. So that's how we sort of form that triangular loop. And it's just sort of neural network operations, but the details are they're in the code, but they're in the paper as well.
Very nice. I have a question from Mario Kren. How do you think, how do you think about AlphaFold in the light of AGI? In AlphaFold, you hard-coded ingenious human domain knowledge to overcome the scarcity of training data. And he adds, how could an AGI even hypothetically solve the protein folding problem without starting from hard-coded domain knowledge?
Right. I mean, that's, yes. We think a system, if we have AGI, it should be able to do anything that we could do in some sense. So, but how it would do it, then it's hard to begin to imagine. But here, yes, what we're doing is this very limited machine learning or very narrow AI where we're building a really dedicated solution and building on years of work, like all this work about, what the physical constraints are between amino acids and this evolutionary knowledge that's been built up over many years to incorporate into our model, and then what the inductive biases are and how to represent them in a network. Yes, it's been a very sort of slow incremental process, and with lots of dead ends where we've tried things and they haven't worked. Yes, it's very difficult to imagine how an AGI would solve that, but it seems like it ought to be able to.
Yeah. Well, intriguing answer and intriguing question, actually. A question from Harish Ritish. I hope I'm not butchering your name, Harish. Apologies if I did. Hi, Andrew. Related to your 2021 genomics paper, are there any works that directly predict TFBS rather than inferring them directly from the network?
So there is work on this is transcription factor binding sites. So these are the proteins that bind to DNA to sort of label which bits of DNA should be turned into proteins. There is work on that. I'm not very familiar with this, but it's something that, you know, we're interested in, that ultimately, you know, if you can predict protein structure, you should be able to predict protein and DNA structure, and therefore we should be able to sort of understand the compatibility between a protein and a given DNA sequence. You know, the geometry of DNA is quite restricted, but these transcription factor binding sites are sort of reading which particular nucleotides are in the DNA. Giga Avsek, who led the work on a genomics paper, he's done previous work on you know, predicting the motifs based on experimental data. But I think ultimately we should be able to come up with a physical model that looks at a protein and can tell you which bits of DNA it would bind to. But I couldn't point to any particular references for that. And I know people are thinking about that, but I don't know of any published work specifically.
Very nice. Thank you very much for your great answer. Two more questions from Azar Abbas. Hi, very excellent job. Kindly explain the term per residue count of non-GAP amino acids in pre-cited MSA.
So I guess that's predicted MSA.
Predicted. Yeah.
I'm not exactly sure what this refers to. I think this is a sort of a technical detail, not something I've specifically talked about.
Fair enough. Fair enough. Then maybe, Asa, if you want to rephrase, feel free to put in the chat. Last question from Andreas Meyer. Very interesting presentation. Is the training data for LDDT prediction the same as for the actual structural prediction? If yes, why do you think there is no underestimation of the errors? And in bracket, assuming there might be at least some slight overfitting in the training data for structural prediction.
Yeah, so that's something that, you know, we've been sort of concerned about, and a lot of people have expressed surprise that it does work out to be calibrated. I mean, basically the data it is trained on is we have these ground truth structures, and then as we go through training, we produce a lot of wrong structures, but we can measure how well they fit the truth, and that's obviously how we generate our losses to train the neural network. But it also, those differences are the objective, the target for this LDDT. It's predicting how accurate is this bit of the structure. So it sees a lot of really bad structures early on in training, and then it starts to see good structures later on in training. And yes, it remains calibrated, but I think, you know, the dynamics of why it's stable and doesn't overfit horribly is, I don't really understand that. I think that, yes, we've been lucky. I mean, a lot of people have worked on these sort of predictions of confidence metrics that said, yeah, you shouldn't have expected this to work, but it seems to.
But it did. Well, Andrew, I think thank you very much for taking your time and going through all the questions there. Thank you again for an amazing talk. Actually, to everyone, it would be great if, I think we'll send you a little survey. If you could fill it out, your feedback is important to us. Thanks very much, everyone, and I wish you a nice evening.